the first talk. So, as I mentioned, I'm going to be talking about our experience in the Australian Epilepsy Project delivering language fMRI at scale. And I have no disclosures. So language fMRI refers to the non-invasive visualization of language-related grey matter functions and resolving this at the individual participant level. So I've lifted this definition from a position paper or a consensus paper put out by the OHBM community earlier this year. And so there's elements of what I'll talk about today that overlap with what's in that position paper. So it's worth a look. Full disclosure, I've got nothing to do with that paper though. So language fMRI, it's generally undertaken because there's a clinical need to know something about where language is localized and lateralized in a particular individual. So why might we need to do this in epilepsy, the context in which I'm working? So there's a number of reasons. First of all, it can help with making sense of seizure semiology. So if we've got patients presenting with aphasic features or dysphasic features during their seizures, it's helpful to know how language is organized in their brain, where it's organized. It's also well known that functional systems like language can be altered in people with epilepsy, especially in the context of things like malformations of brain development or long-standing lesions. So we can't assume the normal rules of organization apply. And then also, it's useful for surgical planning. So if we're going to go in and implant electrodes to try and understand the seizure networks in someone, it's useful to know where our electrodes are going to be relative to language functions. And also, if we're actually proposing a resection to deal with epilepsy, what kind of risk might that pose to things like language function or memory function? So language fMRI in the clinical context, it's essentially an N of 1 investigation. And while I'm not going to go through all the details here, the point I'm trying to make is that it's a complex operation. This involves a multidisciplinary team. Many specializations have to come together to achieve this, to acquire the data, to process the data, analyze the data, and make sense of the data. And it's a time and a labor-intensive process. And so this, begs the question, how can we deliver language fMRI at scale? So in a project like the Australian Epilepsy Project, where we're trying to deliver this nationally, this is a fundamental problem, how to make this accessible at sites across the country. So I keep talking about this Australian Epilepsy Project. What is it? So it's a federally funded national scale project that's aiming to change the way that healthcare management of epilepsy occurs in the health system. And its broad aims are to develop epilepsy-specific outcome prediction and decision support tools for use by clinicians. And it aims to do this by acquiring uniform multimodal data sets in 4,000 plus participants from around the country. And so the way the project works is treating neurologists refer in patients into the project, and then we collect at baseline, we collect cognitive data, we collect imaging data, and we collect genetics data, and then we synthesize all that information back into a report that then is fed back to the referring clinician, and then the clinician can use that to inform their treatment decisions as they work with their patients. And as part of that imaging protocol, all participants in the project, they undergo a language fMRI study. And so now that we're running at scale, this means that we can be reporting 20 plus language fMRIs a week. And so this is the problem we need to address. How do we deliver this? So what's our approach to language fMRI in the AEP? So the first point to note is that language fMRI is but one part of a much larger epilepsy-directed imaging protocol that's occurring in the AEP. So this means that time is at a premium for us. And so we're aiming here to acquire language fMRI within a 3-minute window. So we don't have a lot of time to work with, given all the scans we have to collect. So to cut to the chase, our decision was to adopt a non-word rhyming paradigm. And so in this paradigm, we have an active condition where we're contrasting, the participant has to make a decision about whether two non-words, would they rhyme if they were to be pronounced aloud? And then we contrast this with a baseline condition where they're judging patterns of forwards and backslashes, do those patterns match or not? And so this is all done silently, there's no speaking in the scanner, and the responses are recorded via button box. And the considerations, I'll just go into this briefly, but the considerations that led us to selection of this paradigm is, first of all, it shows good recruitment across key hubs of the language system. So we can see in inferior frontal cortex, this is random effects analysis on a set of healthy controls. And we can see there's activation in the inferior frontal region near Broca's, in the posterior temporal region, proximal to so-called Wernicke's area, and then down in occipital temporal cortex in the fusiform system for reading. So these are key areas that we know are vulnerabilities for surgery, for instance. Another reason is with this paradigm, you get a behavioural measure, so you've got an index of how people are going in when they're trying to perform this task. So how well do they understand what they're doing? It's also a relatively simple paradigm. It's easy to explain to people, it's easy to understand, and there's a minimal level of language function that's required. So it's well suited to the kind of cohort that we're working with. It's also brief. We've shown in previous work that you can generate reliable language activation maps with as little as 3 minutes of data, which is one of our key limiting factors. Now, one of the logistical issues here is how do you train people for this when you're acquiring images from facilities all across the country. So the approach that we've adopted is, you'll recall I said that we do a cognitive assessment for all participants that come into the project, and so we do that cognitive assessment via telehealth over a video call. And we've done this using our own bespoke software that we created for the purposes of the project, and we've been able to build into that software a version of the task that they will see in the scanner, so they have a practice of it. We can instruct them in it, we can make sure they understand what's going on, and they complete this before their visit to the MR facility. So people are familiar with what to expect before they get to the scanner, and then when they arrive at the scanner, just prior to the sequence, or we start the language fMRI paradigm with some slides that serve as reminders as to what they're about to do in the scanner. Now, on this slide, so the image is obviously acquired locally at each MR facility, and in brief, it's a block design task. It takes 3 minutes to administer, and our acquisition is based on a 900 millisecond TR using a multi-band, multi-echo sequence. And then once that data is acquired, it's sent up via the cloud where it's processed centrally using Amazon Web Services. And so all of our processing is done centrally. So this includes the processing of the images, the analysis of the images, the generation of figures that we're going to use in our subsequent reporting back to the clinician, and also our interpretation of the study itself. So here's some figures that show, and right at the start of the project, how we were reporting back language fMRI on our individual cases. And so you can see here that we include a surface overlay of the activation on the person's own underlying T1 image. Then we show axial slices shown on the underlying EPI data through which it was acquired. This is in own space again, roughly aligned to MNI. And then we also include a language laterality curve. And so for those not familiar with the laterality curve, the way you acquire this is you take the underlying activation image and then you threshold it at different thresholds. And at each threshold, you compare how much activation there is in the left regions of interest versus subtract off the number of voxels that are active in the right and normalize them by their sum. So this basically gets you a result where values near +1 indicate fully left lateralized activation, values near -1, fully right lateralized, and values near 0, it's bilateral. So we can trace this out as we adjust the threshold, and if we plot it as the number of active voxels, we get an indication of how laterality is expressed in those regions. So we feed back those types of images in the report, but the other key piece is the interpretation on those images. And so I've given a little example down here in the bottom right, and the way that's done is someone who's experienced in interpreting language fMRIs, they'll put together a report that extracts some of the key information about that image, so there's an interpretation given back with the base data to the referring clinician. So what's hopefully is clear from this is that while there's elements of this pipeline like the image processing, the image analysis, the figure generation that can all be done centrally and automatedly, so that's readily scalable, the interpretation is really a bottleneck here to delivering a high throughput of reports per week. So how can we make this interpretation element scalable? So when we sat down and thought about how to achieve this, and it really, we think there's two primary issues that you need to address. So one is you need a model of what does typical look like. What's the normal behavior you'd expect in the normal population, and a representation of a model of expected behavior. And then the second thing is you need some kind of measure, an objective measure of departure from what's typical. So when you get a given activation image coming in, you can answer the question, how atypical is this particular case? And so our approach to dealing with this was to use regional laterality curves to try and address these two questions. So in our previous work, we had shown that you get strong lateralizing information out of this inferior frontal region, lateral temporal region, and occipitotemporal cortex out of the paradigm that we're using here. So we're focusing, and I also mentioned tumor work and things like that has shown that these are key vulnerability areas in terms of estimating risk. So we operationalize this by collecting a set of we've got 154 healthy controls that we normalize their images to the cerebra symmetric brain space and did a random effects analysis on that. And then we intersected the neuroanatomical parcellations of the Cerebra atlas with that random effects map. And so we draw out 3 ROIs. We've got the neuroanatomical parcellation of inferior frontal cortex intersected with the RFX, the parcellation of superior temporal cortex intersected with the random effects, and also posterior fusiform and inferior temporal cortex intersected with the random effects. And then we take those ROIs on the left, make them symmetric over on the right, and then we can calculate regional laterality. And so now we need to characterize what's the normal pattern that we expect in each ROI. And so we take the same 154 healthy volunteers and we generate laterality curves in each ROI. And then our first step is we discard the 5% most atypical curves. So this is going to catch the small proportion of controls who've got right lateralized language, and it leaves us with the modal type of behavior that you see, which is this distribution of left lateralized laterality curves. Then, at each value on the x-axis here, the number of active voxels, we look at the distribution of the laterality curves, and we test, is that distribution normal? So is it statistically well behaved? And then we go out until we find this region in which the laterality curves are well behaved statistically, in a sense of a normal distribution, and we can use this to define a lower bound outside of which things would be considered atypical. And therefore, any new activation image that we come across, we can run it against this reference set, and we can classify, does it fall within this expected range of typical behavior, i.e. it looks like normal left lateralized language in this region, or does the curve traverse outside of that region, and therefore it's flagging that something unusual is going on here, and we need to do a manual review to interpret that map. So we repeat this process across each of our regions of interest. So I'm showing them here for the inferior frontal region, the lateral temporal region, and this fusiform inferior temporal region. And so here's some examples of the type of behavior we get. So this first one, this would be flagged through this automated process as a typical left lateralized language pattern. So if we look at the axial slices, this is in M&I space now. You can see down in these so-called language slices, strong left lateralization in prototypical regions. The surface peel reveals the same thing. And if we look at these blue curves of this individual's laterality curve, it sits comfortably in the normal band. So it's classified as normal left lateralized language. So we can summarize this in this key figure, which is the cerebrotemporal space showing the ROIs on it. We report the laterality values within the ROIs, and then we've got a summary which basically says it's left dominant in each of these ROIs. Now here's a case where things come up as atypical. And so this is flagged as something's different about this case. We need to actually review it and make sure we understand what's going on and provide an interpretation on that. And so it's immediately apparent when you look at the axial slices that in this case we've got right lateralized language. You can see this on the surface peels as well. And it clearly comes up as this right dominant pattern on the laterality curve. So that's how we've reviewed this case. We've understood what's going on. We can summarize it using our summary image, and then we add to that a manually derived interpretation of what's going on, what we think the salient information is, and that goes back in the report to the referrer. So we introduced this process at the start of this year, and so to date we've analysed 271 language studies, and we've been monitoring how it's behaving over this time. And to date, we're flagging roughly 30% of cases get flagged for manual review, which means roughly 70% are going through automated processing. So this has greatly facilitated our capacity to scale things up and deliver these reports. I will note, though, that we're in our experience, we're getting roughly 2% of studies are unsuccessful. So here's an example of an unsuccessful study. So this is thresholded at P001, and you can see that there's basically no meaningful language activation in this map. And when we pass it through the automated pipeline, if we look at the unthresholded image, you can see it's just kind of a noise background of weak activation and deactivation with some kind of unusual clusters up near right motor cortex and down near mesial temporal lobe on the right. And so what happens when this comes through our automated classifier is because of this random distribution of noise, the curves tend to come out bilateral, and so it gets flagged because it's outside of this normal behavior band. So this leads us to review it, and then we can feed back in the report that this is an unsuccessful study. If lateralizing or localizing information is needed, we'd recommend some other types of investigations to explore that. And so I'll end by, showing how this is actually useful in individual cases. So here's an example that was referred into the project. It's someone who had refractory focal epilepsy, and they came in, they underwent the AEP imaging protocol, and hopefully you can see it on this slide, but this uncovered this unusual sulcus in the left medial frontal cortex, and this proved to be a large extensive region of photocortical dysplasia. So the good news is we've now got a good idea where this epilepsy is coming from. The bad news is this is close to prototypical language country, so there's risks with a surgical intervention, especially with large tissue like this. But as part of the AEP, they had their language study done, and we can see here straight away that this person is right lateralized for language. So this dramatically changes the calculus about what's the risk profile, what's the tolerability of surgery, how extensive can we go with surgery. So this is an example of how this is actually meaningfully impacting things for the patients that are coming through the AEP. So that's a good point for me to hand over to Lee. So as Lee's coming up, I'm just going to acknowledge the many people in the team that have assisted with this work. And I'll do a quick shout out to some of our work, some of our scientific work that's emerged from this as well, looking at connectivity during language processing, published earlier this year. And you can go and visit Vicky He's poster, a PhD student, poster number 1400, which will be presented later in the week. So thanks everyone, and I'll hand over to Lee.